Hello students. Today in this session we are going to see the practical part. Under the practical part, the paper, the biodiversity, microbes, algae, fungi and archegonata. In our last session we have seen regarding our syllabus part. What we are going to deal in this academic that we seen in our previous session. In this session we are going to see regarding practical part. Here, there are few key terms which we frequently see in our practical part, such as TS, LS, VS, VM, TLS, and RLS, which means TS refers to transverse section, LS refers to longitudinal section, VS refers to vertical section, WM refers to wet mount, TLS refers to tangential longitudinal section, where RLS refers to radial longitudinal section. These keys are important to know the practical part. We see frequently these terms. Here, the practical part under the paper code BOT DSCT P1.1, that is practical part paper 1. The marks allotted is 40 and IA will be given 10 marks. We are going to perform the practical for 40 marks and IA is allotted for 10 marks. The teaching hour for practical is 3 hours per week and the credits for this practical is 1. The total your botany subject credits for 4. That is 3 for theory and 1 for practical. That is botany credits for 4 points. Here, under the first practical, with the help of EMS or models of viruses, we are going to study T phase and TMV, that is tobacco mosaic virus. Next, with using the diagrams or photographs, we are going to study the lytic and lysogenic cycle of viruses. In second, the types of bacteria from temporary or permanent slides or photographs. Either using one, we are going to study EM bacteria, binary fission, conjugation and structure of root nodule. Next comes the gram staining. Under the gram staining, we are going to study double stainings, that is gram staining. Next comes the study of vegetative and reproductive structures of volvox and nostoc using electron micrographs. Next, the Oregonium, Vachera and Ectocarpus and Protactospermum through temporary preparations and permanent slides. Using the permanent slides of temporary preparation, we are going to study Oregonium, Vachera, Ectocarpus and Protactospermum. Next, Rhizopus and Penicillium. Here we are going to study the asexual stage from temporary mounts and sexual structures to permanent slides. That is asexual stage and sexual stage that we see in Rhizopus and in Penicillium. Here we are going to mount to study the asexual stage of Rhizopus and Penicillium. Next come Cercospora. With the help of specimen or with the help of photograph, we are going to tease the mount. With the help of specimen, we are going to study in detail with the help of photographs. In the case of Cercospora. In case of Puccinia, here the herbarium species of black stem rust of wheat and infected berry leaves. Next, here the section or we tease the mounts of spores on wheat and permanent slides of both the hosts. Here we tease the specimen where we get the infected spores in the infected part of the plant. Here with the teasing material, we will study the types of spores present in the Puccinia. Next, Agaricus. Here the specimens of button stage and full grown mushrooms. Sectioning of gills of Agaricus. Here we are going to take the section of Agaricus and we will study the types of spores present in Agaricus and the structure, the gills, the nature of gills in Agaricus. Next comes Lichens, here the study of growth forms of lichens. Here what are lichens? As I stated before, these are the mutual combination of algal and fungal members. Those are called lichens. Next comes mycorrhiza. Here two types, that is ectomycorrhiza and endomycorrhiza. With the help of photographs, we are going to study regarding ectomycorrhiza and endomycorrhiza. Next comes Markantia. In the case of Markantia, we are going to study the morphology of thallus. Next, the wet mount of rhizoids and scales, the vertical section of thallus through gamma cup and wet mount of gamming. Here we are going to study with the help of temporary slides. We are going to prepare these slides. With the help of these slides, we are going to study. Next, VS antheridophores, VS the vertical section of antheridophores and archegoniophore. Next, LS, that is longitudinal section of sporophyte with permanent slides. We are going to study the LS of sporophyte with permanent slides. Here in Funaria, the morphology, the wet mount of leaf, rhizoids, operculum, peristome, annulus and spores with the help of temporary slides, and permanent slides showing antheridial and archegonial heads, LS of capsule and protonema. This we study in case of Funaria. Next, Selaginella. Under Selaginella morphology, wet mount of leaf with ligule, transverse section of stem and wet mount of strobilus. Next, the wet mount of microsporophyll and megasporophyll. Here we are going to prepare, that is the temporary slides. Next, with the help of permanent slides, we are going to study the LS of strobilus. This is in the case of Selaginella. Next, in Equisetum morphology, the transverse of internode, the longitudinal section of strobilus, the transverse section of strobilus, wet mount of sporangiospore and the wet mount of spores, that is wet and dry, using the temporary slides. Next, the TS of rhizome with the help of permanent slide. We are going to see in the case of Equisetum. Next, in Pteris, we are going to study the morphology, the transverse section of rachis, the vertical section of sporophyll, the wet mount of sporangium, the wet mount of spores using temporary slides, whereas TS of rhizome, the wet mount of prothallus with sex organs and young sporophyte using permanent slides in the case of Pteris. Next, in Cycas, the morphology, that is the coralloid roots, bulbil leaf. In morphology, the transverse section of coralloid root, transverse section of rachis and vertical section of leaflet, the vertical section of microsporophyll, the wet mount of spores, that is using temporary slides, and the longitudinal section of ovule and the transverse section of fruit using the permanent slides. Here, in last case, that is in the pinus, we are going to study the morphology. Under the morphology, long and drop shoots, the wet mount of drop shoots, the male and female. Here both the male and female, we are going to study the wet mount of drop shoots and the long shoots. Next, the wet mount, drop shoots, the transverse section of needle in pinus, the transverse section of stem, the longitudinal section or the transverse section of male cone, the wet mount of microsporophyll, the wet mount of microspores using temporary slides. We are going to study with the help of temporary slides, and the longitudinal section of female cone and the tangential longitudinal section and radial longitudinal section of stem using the permanent slides. In last case, that is in the pinus, the morphology. Next, the wet mount of drop shoot, tears of needle, tears of stem, LS or tears of male cone, the wet mount of microsporophyll, the wet mount of microspores using the temporary slides, using the permanent slides, we are going to study the LS of female cone, the TLS and RLS of stem. Next, the study tour, 2 days for your study purpose. Here we will go for study tour, where we have to get the report of the study tour regarding what you have studied in your tour section. This is regarding your practical part. That is, we are going to deal 50 marks for practical, where in 50, 40 for your practical part, where we are going to perform for the 40 marks, and 10 marks is allotted for IA, which credits 1 for practical, and the theory credits is 3. Total the credit points for botany is 4. I hope you got clear with practical section. Thank you.